This is our third formal lab this semester called Absorbance and Fluorescence. It is created by Lenny Bills, Hannah Boardman, and Michael Moran. In this lab, there were a few research questions conducted. One being, what kind of dye is used in this lab if we compare it to the known absorbance spectra? Another question was, what is the concentration of this dye? How does the wavelength of fluorescence dye relate in terms of absorption and emission? And lastly, why would the fluorescein emit a different spectra than what it absorbed earlier? First, we're going to identify what some of the terminology means in this lab. Reflection is the color that is not absorbed by an object. This is the color our eyes see when we look at an object. For example, an object that appears green reflects green wavelengths. However, on the other hand, absorbance is the complete opposite. These are the colors that are not reflected by the object. They're all the colors our eyes do not see when we look at an object. Fluorescence is the emission of light from an object when excited by light or energy.
Some models and equations that we used during the lab were Beer's law and the intensity equation. Beer's law we used to find concentration, and intensity equation we used the intensities to find absorption. And then between the two setups, we used, again, optical cable, a computer, a spectrometer, a flashlight, and our veil of dye or fluorescein. However, the only difference between them is that for the fluorescein, the light has to be perpendicular to our optical cable, vice versa, to properly measure the emission.
In order to perform this experiment, we first had to set up the spectrophotometer to show absorbed light as shown in the model slide, making sure to measure the distance between the cuvette and the meter where the graph was at its optimal peak. We repeated this for a cuvette filled with water. We exported this data to an Excel sheet and calculated the absorbance by using the intensity equation. We then used Beer's law to find the concentration. We compared our plot of absorption to the known absorption peaks given to determine our kind of dye. Then we found absorption values for the fluorescein the same way. Next, we set up the spectrophotometer to measure emissions of the fluorescein as shown in the model. This was done by having the optical cable perpendicular to the flashlight. For our calculations, we first found the absorbance of the dye by using the intensity equation. We took the log of I naught divided by I. We used the I values that were at the peaks of the absorption graph. Following this, we used Beer's law to find the concentration of the dye. We did this by rearranging the equation to solve for concentration. So we divided the absorption found from the intensity equation by the product of the molar absorptivity constant given and the distance we measured between the cuvette and the optical meter. We found that our dye ended up matching the Sautel dye. We figured this out by comparing the peaks of absorption and seeing that both had their peaks at 500 nanometers. This made sense because our dye appeared orange, so it reflected orange light, which can also be shown from the absorbance graph having a trough at 600 nanometers, where the orange would be. We found the concentration of our dye to be 1.22 times 10 to the negative 8th moles per liter. For fluorescein, we found that the absorbance peaks were at around 530 nanometers and the emission peaks were at around 550 nanometers. It makes sense that these are different because the energy of emission is less than the energy of absorbance.
So now let's talk about the scientific reasoning behind this lab. First of all, we used Beer's law because it states that absorbance rates are directly proportional to our concentration of dye. This was seen in our calculations as the two graphs had a positive correlation to one another. Next, with the fluorescein, the absorbed and released wavelengths of energy differ from one another because the energy must be conserved. Energy and wavelength are inversely related to one another, allowing for a longer wavelength to release and or absorb less energy.
In the discussion portion, we compare the wavelengths between absorbance and emission patterns in addition to what each spectra displays. To start off with, an absorption spectra shows wavelengths that are absorbed from our dye, while the fluorescence spectra shows wavelengths that are fluoresced or emitted from our fluorescein. We can use the absorption spectra to identify unknown compounds through comparison with known absorption spectrums, while we use the fluorescence spectra to help recognize different types of cancer. When fluorescein absorbs energy at a certain wavelength, it reaches the excited state. The fluorescein will then resonate or bump into other molecules where kinetic energy is released. Since energy is released here, the potential energy released from the fluorescein to the ground state is less than what it was absorbed at in the beginning. Although these different wavelengths were used in the experiment, the energy is still conserved. As mentioned in Part A, kinetic energy was released as fluorescein bumped into other molecules. However, since the total energy equals potential plus kinetic, the potential energy released must be less when it reaches the ground state in order to conserve the energy. We recorded our measurement in the dark so that the fluorescein didn't decompose when it was exposed to natural light. Moreover, the fiber optic cable and flashlight were kept perpendicular so that I would only measure the light that was emitted instead of the light that was absorbed.
For this lab, there could have been possible errors and limitations. One could have been the angle between the light and the optical cable was not perfectly perpendicular. Next, the room could have been not completely dark, so there could have been measurements of light from other sources. Next, the optical cable was not properly calibrated, so either it was getting more or less light between each graph, or the number of readings between each graph were not the same. And finally, the fluorescein could have been exposed to some light between experiments, so the amount of light being emitted could have been off.